So we'll start from just the picture of, a pair of reads that overlap over some segment, but then they each have portions that do not overlap because they start and end at different points on the genomic coordinate. So now let's project that sort of onto x and y axes. So let's imagine then that this lower sequence, maybe that's our x dimension, and then our other sequence, we can imagine that being our y dimension. So so now this region of overlap would basically be along here in our x sequence, and going from beginning to end of our y sequence, it would be sort of over the corresponding end of our y sequence. So I'll explicitly write that's our x sequence, that's our y sequence, so there's our x sequence, our y sequence. So according to this picture then, we expect overlap basically between those two little segments. So what will that look like? Right, so we're basically looking for a diagonal-like path going across that region. There could be little insertions or deletions in there, but basically a path approximately like that. So one way of saying that is that we're looking for a path with a certain constraint on it. We've talked about two different kinds of constraints before. For the global, what was the constraint? Right, so for global, it was you had to start there and you had to end there. And for local, what was it? Yeah, basically, you're pretty much allowed to start and end, anywhere you like inside the matrix, as long as it gives a maximal score. So sort of unconstrained in the local case. So what would you propose then for constraints on the overlap alignment path? Hopefully the picture kind of helps suggest. Absolutely, absolutely. So it's sort of like, we extended the global starting point to basically be anywhere on one edge going to anywhere on another edge. And just to be completely general about this, of course, that also applies to the other edges, right? Because we don't know what order the two sequences came from. So, for example, we could have an overlap like that would basically be like aligning that portion of my x sequence to the first portion of my y sequence, like that, right? So, so the proposed constraint then would be a path from one external edge across to the adjacent external edge of the matrix. Make sense? Question? Okay, so you're talking about an alignment that would look like there's x, and you're saying some middle portion of x, then aligning against some other middle portion of y, like that. So I can make up scenarios from other kinds of molecular biology experiments where that might make sense. but specifically for short read sequencing, if these really are short reads from the same region of the genome, then they should overlap from at least one end into where the other one ends. If there's extra stuff stuck on the end, that's most likely to imply that we're looking at some kind of repetitive region that occurs in multiple locations in the genome adjacent to other sequences that are different. So the implication would be that this portion that's aligning nicely would be a repeat that's found in two different places of the genome. So the repeat part aligns, but the parts on either side of it don't, because they come from different places in the genome. Another possibility would be that there's been an error in the experimental library preparation, so that there's extra vector sequence or something stuck on the ends, but that's not supposed to happen according to the library design. So we don't, if the data is bad, we don't, we sort of don't want to see it, at least according to the standard. Basically, if the stuff is working, then what we want are end-to-end alignments.
We just want to follow up. Maybe I'm understanding exactly what we're looking for. So what if the cost, let's say about 150 is we actually find the match is say 5,000 now, and then Y I added 100 nucleotides of the region, and then say for that, right? And X added 150.
Okay, well, the first key starting assumption that maybe I should emphasize was we were saying that all of our short reads are from the same genomic sequence. So we're not talking about an X and Y that are separated by some kind of big long evolutionary process. We're talking about resequencing basically the same thing over and over again on different overlapping short reads in order to build up an assembly of what the original genomic sequence was. So you're right. This is why we want to use different kinds of alignment constraints for different kinds of source data. So if you were talking about something that had these kind of interesting evolutionary events like insertion or transposition of blocks of sequence, and we want to align taking those events into account, then we want to use a different set of constraints. But here, where we're just trying to assemble one genome from a bunch of overlapping reads, then we probably want to use this constraint. Did I answer the question? Okay. Other questions? Okay. So basically an edge-to-edge alignment. You've already seen local alignment. It's another variant of this where basically we're unconstrained in our start and end points. One can play additional kinds of games like this with the alignment start and end, but I think you get the idea.